Chapter 3, Structure and Function of Cells, Screencast 2. Now, the primary focus of this screencast is going to be the plasma membrane and how it surrounds the cell. So a couple of things I really want to reinforce here, okay? So one of the big jobs that the cell membrane is doing is it's keeping the cell away from its external environment. I want us to think of it as a fence, right? It's actually holding together the container of the cell itself. Now, there are some really special characteristics. And one thing that is really important is the cell membrane is what we call selectively permeable. And what that means, some substances will enter the cell and exit the cell, but others can't. Probably the most important determinant as to what will freely move into and out of the cell is solubility. So only things that are lipid soluble can easily enter and exit the cell. Now, that's a problem because if we look at some of the biological molecules that we talked about in the previous chapter, a lot of those are polar, which means they can't diffuse through. So carbohydrates such as glucose, amino acids, and all sorts of other things are not able to simply cross through that. So there has to be a solution. How is that going to happen? And that's going to be one of the things that we address. The other thing here, when we talk about the plasma membrane, it's actually allowing transfer of information between the environment and the cell itself. Now, here is an image of the phospholipid bilayer. So remember, phospholipids were introduced as a type of lipid in the previous chapter, and they're very interesting because, once again, they have both a polar and nonpolar component. The head is polar, the tail is nonpolar. Now, the phospholipid bilayer indicates that these phospholipids are arranged tail to tail, with a polar head always facing an aqueous environment. So there's the polar head there. There's extracellular fluid. Here's the other polar head, and here we have intracellular fluid. So that's inside the cell, and there is fluid here. That's important, okay? And other things that we're going to find are cholesterol, and that's kind of built into the cell membrane, and it actually results in a slight degree of rigidity. Not a lot, because we are going to refer to the cell membrane as overall non-rigid and a fluid mosaic. Now, one of the things that I want to point out here is look what I got going on here. Purple proteins, all right? So proteins are going to be purple. And look what they're doing. They're going all the way across from one side of the membrane to the other. Here, this is a receptor protein. I'm not going to spend a lot of time talking about this. We're going to come back to this when we do the endocrine system, chemical messengers, neurotransmitters, and so on. Okay. Now here, this is important. This is a protein channel. This actually happens to be a channel that's always open. So this is going to be specific to ions. So ions typically will travel through a channel. Here, I have a gated ion channel, and we'll talk a little bit later about what causes this gate to open or close and allow ions to move through down their gradient. Here we actually have a transport protein. So this is something that we'll see a little bit later, okay? And one thing that we don't see here are probably pumps, okay? So remember how I said there needs to be a solution so things that are not lipid soluble can actually get into and out of the cell? These proteins play that role, okay? So that's kind of the solution overall. Now also we have these carbohydrate groups, and what they are primarily doing is they're actually identifying this cell as self. So the immune system knows that this actually kind of belongs in this body. Okay, so really important here, these major characteristics of the cell membrane. Now, I do recommend that you take some time and review this animation because it really is important focusing on the overall structure of the membrane, and sometimes it's really nice to have a video clip so that you can actually get a better handle on that. So that's what you're going to find here in this animation. Now, I've already talked about this concept of transport, and when we talk about transport, we're going to break it down primarily into two general categories. Okay, I really don't want to talk about bulk transport because I think that's kind of confusing. We're going to hit that a little bit later. But I want us to think of passive transport versus active transport. Now, passive versus active basically means, does the cell need to actually expend energy? All right, so passive transport, the cell does not expend energy. And the examples that we typically use of passive transport are going to be diffusion and osmosis. Okay? The terms that we're going to see here is down the gradient. Okay? Now, active transport, the cell must expend energy, and the protein involved in active transport is going to be a pump. Now, what's going to happen in this situation is that pump is going to move something against its gradient, and kind of imagine pushing against something, right? You have to work hard to push against it, okay? So that's what the cell has to do. It's actually pushing something against its gradient, and there's a cost associated with that. Generally, when we talk about active transport, that cost is going to be ATP, but we'll see something else also. Now, bulk transport, we're going to talk a little bit about later, endocytosis and exocytosis, and we saw some examples of that in the previous screencast. Now, take a look at this particular diagram here. Okay, there's a couple really important points here. I have this dye, all right? So this dye is pretty much sitting here, and look what happens as time goes by. Notice the dye is dispersing. That's diffusion. So it's basically going from an area of high concentration to an area of less concentration, and eventually some equilibrium is going to be attained. But notice what's happening here. Okay, it's not just going in one direction. Okay, so that dye is moving up, it's moving down. If this were a wider container, it would be moving across, okay, until an equilibrium is actually reached. This is a perfect example of diffusion, and no energy is being spent here, okay. Another real example is if we look at diffusion. I spent some time living out west, and I'm really concerned right now because of the forest fires. Okay, we're already starting to see the smoke from the forest fire, or the impact of the smoke and the ash here. I've got friends that are photographers, and they've been posting these fabulous pictures of sunsets. Now, that extra smoke has created a haze which enhances the colors of the sunset. So that's a perfect example of diffusion, because once again, that's all located on the West Coast, and here we are in Western New York experiencing the consequences of those forest fires. Another example, kind of imagine back in the day when a restaurant would have a smoking area, right? So smokers would be on one area, non-smokers in the other, but smoke never stayed in just the smoking area, right? There's an area of high concentration, and then it works its way until the equilibrium is actually reached. So those are all really examples of diffusion. So really the definition of diffusion is when something, typically a particle, is moving from an area of high concentration to an area of low concentration. And eventually it's going to reach what we call in a dynamic equilibrium. So things aren't moving, they're just in an equilibrium with no net movement to either area. Okay, so as I said before, the concentration is high to low, and we call that moving down the gradient. Now, osmosis is the passive movement of water. In the previous chapter, we talked about the special properties of water and so on. So water gets its own term to describe how it moves passively. Okay, so water can move across a selectively permeable membrane, and this gets a little confusing. So the definition here is water moves from an area of low solute to an area of high solute. Okay, that sounds a little confusing to me, so I want you to keep this saying in mind that describes osmosis. Okay, water follows solute if it can. All right, so water follows solute if it can, or we can rephrase that as solute sucks. Okay, so that solute is going to pull water towards it. So we can actually see that here. So notice this experimental setup of a U-tube. So in the middle, I have a selectively permeable membrane. Now glucose is too big, so it can't move through, okay? Otherwise, we would see it diffuse through, and there would be glucose particles here, but that's not the case, okay? So I've got glucose here, and it's too big to get through this selectively permeable membrane, and here's my water. Now remember, water follows solute if it can, so that glucose is actually exerting a suckage force, and look what happens, okay? So we see a much greater difference, okay, in the size of where the water is on this side of the tube and an increase over here. And what that means is that water has moved towards the solute because it can, okay? So one of the things that we see here is this term osmotic pressure. I think osmotic pressure is kind of a confusing term, and it's not a term that I use pretty regularly, okay? Osmotic pressure is the fluid pressure required to exactly oppose osmosis. So what would I have to do to make these tubes, okay, the water equal again on either side, okay? I'm more interested that you understand this concept of osmosis, and water freely moves, if it can, where the solute is. Now, here we look at some examples of diffusion, okay? So these are all diffusion, and the cell does not need to expend any energy for this to take place, all right? So here we're outside the cell, and we have now something that's lipid soluble, all right? So that can move easily through. Examples of things that are lipid soluble would be our respiratory gases, oxygen, carbon dioxide. If we look at alcohol, okay, so they're going to easily move through the cell membrane down their gradient, and the cell does not have to expend any energy. Okay, the movement is due to molecular movement. So here, let's imagine we have a glucose molecule. Excuse me, this is for an ion. So we're going to talk, this is an ion channel. So we're talking about a charged particle, okay? Charged particles are not water soluble, okay? Now, water is also an example of a polar molecule, and the cell has special pores called aquaporins, which are specific for water to move down its gradient. Okay, so let's kind of take a look at this. So let's kind of imagine we've got an ion channel here, okay? So this ion is basically moving through this particular protein channel, all right? It can't diffuse through because it's not lipid soluble. It's charged, so it's going to go down its gradient from outside to inside. Here, we're going to talk about something such as glucose or amino acid, okay? We're calling this facilitated transport. Okay. Now, facilitated does not mean active, all right? We're still talking about facilitated diffusion, so the cell is not expending any energy. But here, glucose is a polar molecule. It can't move, okay, or diffuse through the cell membrane because it's not lipid soluble. So embedded within the cell membrane, I have these glucose transport molecules. It's still going down its gradient. And so kind of imagine, okay, you got a kind of a revolving door. Okay, doors open and then opens on the other side, and based on the gradient, because there's little to no glucose here, that glucose moves in passively, okay? That's important. Facilitated diffusion is not active transport. The energy to fuel the process is still a concentration gradient. So glucose is still moving down its gradient, okay? Now here, we've got two different types of active transport. In active transport using ATP, sometimes that's referred to as primary active transport, and now you're moving something against its gradient, okay? The proteins here are pumps, okay? And pumps are always moving something against a gradient. If you have a fish tank, think about the filter, right? You need a pump to move water across the filter to pick up the contaminants. All right, so now look what's happening here. I'm moving something against its gradient, and when we talk about primary active transport, okay, one of the questions you want to ask, what's fueling the pump? So primary active transport is when an energy molecule, specific, especially ATP, is used to fuel the pump. We're going to talk about the sodium-potassium pump, which I think is the best example we have of primary active transport. Now here, this is secondary active transport. And remember the question I said, how are we fueling the pump? In this type of transport, quite often it's going to be called sodium linked. And what's happening, sodium is moving down its gradient. Okay? Now, when something is moving down its gradient, there's energy associated with that. So the energy of sodium moving passively fuels the pump to move something else against its gradient. Okay, so here, remember, when we're talking about against the gradient, I want you to think of it as you're going upstream, right? If you're in a stream or a river and you're kayaking or paddling, you got to work to go against the current, don't you? Okay, that's what the cell is doing here. Okay, so the cell is working against a current of substances, okay, that it's moving against, and that requires energy. If I'm going downstream, okay, like passive transport, I'm just going with the flow, right? Okay, I don't really have to expend any energy. Okay, I may just use my paddle to make sure I'm going straight, but I'm just going with the flow. All right. So really important that you can distinguish the differences between active, passive, and also when we talk about active transport, distinguish between primary, which involves the use of ATP, and secondary, which involves the use of some molecule moving down its gradient, and that energy is fueling the pump. Okay. Here, this is a really good animation that I recommend watching, and it's going to pretty much focus on passive and active transport. Now, I talked a little bit about endocytosis versus exocytosis. Now, this is just using a vesicle to move things into and out of the cell. Okay, so here notice I've got a bunch of this stuff and it's creating a invagination in the cell membrane. And notice what happens. It gets deeper and then the membrane kind of closes off and it clips off. Okay, so now that's inside the cell. Exocytosis is the opposite. Okay, but these are both examples of transport that are using vesicles. Okay, and so here I've got the vesicle full of this stuff. It migrates to the cell membrane, kind of opens up, and it spits out the contents. Okay, so endocytosis is getting in, exocytosis is out. A really good example of exocytosis is how neurotransmitters are released by neurons. So this is another animation you can spend some time taking a look at about endocytosis and exocytosis. Now, I'm not going to spend a lot of time talking about here because it doesn't belong here. Okay, this is showing a receptor and a signaling molecule. I want to talk about this when we do neurotransmitters or hormones. So we're not going to really spend a lot of time here. So I won't be asking anything about this. Okay, all this is showing it's a receptor protein in the membrane. Okay, that's really why it kind of fits in this particular chapter. But I want to go into a lot more detail about this later because it's an important mechanism. Okay. Now this looks a little confusing. All right. And this is an image of the sodium potassium pump. Now remember, I talked about the sodium potassium pump as an example of primary active transport. Okay. Now, I want to talk a little bit first. I want to build a story here. Okay. I want us to know that sodium is the primary extracellular ion. All right. So we find sodium outside. All right. So generally, on its own, sodium wants to move into the cell down its gradient. Okay. Potassium is my primary intracellular ion, and the direction that potassium wants to go, if it can passively, is out of the cell. Okay, this is important because now we're talking about a pump. Now, in an active situation, we're going to be pumping sodium out of the cell. And remember, because sodium is the primary extracellular ion, I have to pump against, I'm going up my gradient. Okay? Same thing with potassium. Okay, potassium is my primary intracellular ion, right? So I have to push harder to get it into the cell. Okay, so we need pumps to do that. All right. Now, one of the things that's an issue, if sodium comes into the cell, okay, and potassium has a lot of leak channels, so it's kind of leaving on its own pretty regularly. Okay, so if I've got this positively charged coming in and accumulating, I'm going to change the electrical properties of the cell. Now, what we'll talk a little bit later is what's known as a resting membrane potential. Okay? The inside of the cell is negative with respect to the outside. So if I have these positive charges coming in and they stay there, I'm going to lose that membrane potential. Okay? So I have to have a mechanism that's going to remove or push out sodium. Okay? And once again, I want to maintain that. So I want to make sure that I have more positive charges leaving the cell than coming into the cell. So there's a ratio. Okay? So when this pump is active, it's fueled by potassium. And this pump is very important. It's going to maintain cell volume. It's going to maintain membrane potential. And because it's active transport, this expends a lot of energy. Okay? A big part of our resting metabolic rate is attributed to this mechanism, the sodium-potassium pump. Okay? So I've kind of built a story here. Okay? But now, this is what I want us to remember. We're going to talk about a pump cycle. All right? So for every pump cycle, 3 positively charged sodiums are going to be pushed out of the cell, and 2 positively charged potassium ions are going to be pushed into the cell. Okay? So I have a net loss of 1 positive charge, right? So that's how the cell, 1, is getting rid of those positive charges and maintaining that membrane potential. Okay? So really important concept here. Okay? So we want to kind of keep in mind. And once again, this is primary active transport and fueled by ATP. Now, I'm not going to spend a lot of time here because I think this slide is kind of confusing. And what I want you to know here that one of the big jobs of the sodium-potassium pump is maintaining cell volume. Okay, I'm not going to go beyond that. I actually think the role of maintaining the membrane potential is more important. So we're going to come back to that a little bit later. Now, another set of terms that you kind of want to remember here is this concept of tonicity. And tonicity is basically the relative concentration of solutes in 2 fluids. Okay? In isotonic, the extracellular and intracellular concentration gradients are equal. So what this means, there's no osmotic gradient. Okay? Because remember, water follows solute if it can. Okay? And you maybe have had IV fluids. And one of the things you really hope when they're putting those IV fluids in, it's isotonic saline. Okay? So the fluid they're putting in, okay, through that IV has the same solute concentration as your blood. So you're not going to have an osmotic gradient. Okay? There's not going to be a force that's going to have water moving from one area to another. Okay? So that's isotonic. All right. So now, panel A is showing isotonic. Okay, so we've got 9 grams of salt in 1 liter, and that happens to be the same solute concentration inside of a red blood cell. All right, so there's going to be no net movement of water. Okay, so that cell is going to stay the same size. All right, now, okay, we've doubled it. So I now have 18 grams of salt in a liter. It's a highly concentrated salt situation, okay? So think about what I have now. Where does water go? It goes where the solute is. So the water inside my blood cell is actually going to leave, and that cell is going to shrink. And we can see that here, okay? Now, hypotonic is when here I have water that's solute free, okay? So there's no solute whatsoever in this water. So it's distilled water. The solute, though, is inside of the cell. Where does water go? It goes towards the solute. So water is actually going to move into the cell, and eventually might happen, that cell, that blood cell, will burst. Now, you may have heard of situations related to water toxicity. Okay? That's what's happening here. Someone drinks a large volume of water in a short period of time, okay? And that results in a situation where blood cells will actually start to expand and lyse, okay? So really important concept here. And one of the things that we regulate pretty tightly within our body is plasma solute concentration.